says he's ready. Okay, I guess we can get started. I want to thank everyone for coming to the afternoon session. And the first speaker of the session is Richard Axel, comes here from Columbia University. Richard?
Thank you. I too would like to say that indeed I've watched the Pick Hour for the past 15 years as the Center of Learning and Memory. It really is a privilege, a pleasure, an honor to have the opportunity to celebrate with you this afternoon. Last night I had an opportunity to think and talk about Barbara's contribution. And today I want to briefly talk about Susumu. Susumu has been a remarkable founding leader of the Pick Hour. And it made me think about his enormous resilience as I thought about his career. And so let's consider his resilience. This, is that you? Is the young Susumu as an undergraduate in Kyoto University. He then, after completing his graduate degree, moved to the Salk Institute to work with Renato Del Becco, where he, in fact, took on all of the character of surfing La Jolla. Here's Susumu as a postdoctoral fellow, and in fact, he already presaged, you see the DNA tattoo that he had, presaged DNA rearrangement. He went to Basel, to the Basel Institute of Immunology, and again adopted the character of the Swiss. He constantly went around with this lid and this traditional Swiss garb. And now we see Susumu today in front of what looks like a Tiepolo. A remarkable ability, a chameleon almost. So thank you for this opportunity. This is not a nose. It's a portrayal by the Belgian surrealist René Magritte of his own brain's representation of the external world. It is a vignette of image and reality locked in mutual cancellation. The problem as to how the brain represents the external world is not only a creative force in art, it's central to philosophy, psychology, and neuroscience. And the question of how the brain represents the olfactory world, I'd like to talk to you about today. Now, all organisms have evolved a mechanism to recognize sensory information in the environment, transmit this information to the brain, where it is then processed to create an internal representation of the external world. This internal representation, this map, must then translate stimulus features into meaningful neural information so as to elicit appropriate thoughts and behaviors. In vision, in touch, in vision, touch, and hearing, features central to perception exist in two-dimensional order in the peripheral sense organ, and this order is maintained as the sense organ projects to lower cortex. In olfaction, however, it's difficult to conceive of any feature of this sensorium that exists in two dimensions And there is no odor in the peripheral sense organ, in the nose. And this immediately poses the question as to how olfactory information can be meaningfully represented in the cortex. And so let's consider this problem in the context of the anatomy and molecular anatomy of the olfactory system. Very quickly, let me review for you the peripheral sense organ, the nose. This is the sensory epithelium. In the sensory epithelium are an array of bipolar cells. In this genetically engineered mouse, the cells are making only 1 of 1,000 receptors. Each cell in the sensory epithelium makes only 1 receptor. Neurons expressing a given receptor are randomly distributed within zones of the epithelium. But order is restored as we follow the axons through the skull. So here's the sensory epithelium, the turbinates. Here is the skull. The axons of these randomly distributed neurons making receptor X all converge on a fixed point in the olfactory bulb. 2 important properties about this convergent point known as a glomerulus. First, the neurons expressing different receptors all project to different points in the epithelium, in the olfactory bulb. And finally, this pattern of projections, this apparent map, is stereotyped in all individuals in a species. So we have an anatomic map, and immediately follows from this anatomic map that different odors will activate a different ensemble of receptors, which will activate a combination of glomerular loci in the bulb, such that the quality of an odor should in fact be determinable by spatial patterns of neural activity in the bulb. And in fact, this is true in the earliest of imaging experiments, first done by the late Larry Katz and his colleague, and subsequently repeated in many other laboratories, including our own, show that different odors elicit different spatial patterns of neural activity. So here you have 4 glomeruli that are activated by the smell of banana, and adjacent to it, a single glomerulus activated by the active component of fox urine, as we'll see later, a rare odor that elicits innate behaviors in the mouse. So this stereotypy, in fact, allows us, with a 2-photon microscope, to look down on the olfactory bulb and, from the patterns of neural activity, discern the nature of an odor that an organism has encountered in the external world with a reasonable degree of accuracy, which is quite neat. But the central problem, however, is not solved because the mouse does not have within its brain a 2-photon microscope. So the problem is how the mouse looks down on this pattern of neural activity and discerns the nature of the odor, and from that, an appropriate behavioral response. And so we in the laboratory have begun to follow the projections from individual glomeruli in the bulb to higher cortical centers and subcortical centers that represent odors. So what you see here is a correct drawing by Ramon y Cajal, which delineates the pattern of projections from individual glomeruli. Here you have a glomerulus. The glomerulus receives sensory input, afferent input. Each glomerulus receives about 20 dedicated, dendrites of 20 dedicated projection neurons known as mitral cells. These mitral cells, as we will see, then go up and pentafurcate. They project to 5 major regions of the brain. I apologize for the use of these simple schematics, but I just gave a talk at Harvard. So Dara Siselski, a graduate student, and Bob Datta, now a professor at Harvard, God bless him, worked out a simple way to look at the projections from individual glomeruli. They worked out an iontophoresis procedure in which they could simply iontophoresis dye into a single glomerulus such that that dye would not spread to neighboring glomeruli. And we could demonstrate that quite convincingly. And what you observe when you label a single glomerulus, and so now we're looking at the projections of only 20 mitral cells, gives you a picture of the complexity that we in higher organisms have to deal with. 20 projection neurons gives this pattern of projections in the mouse brain. And what you see are the 5 major areas that receive input from a single glomerulus. Today, I want to talk only about 2 regions. The largest area of the mouse brain and the area that receives the most projections is piriform cortex, or olfactory cortex. And a second area is cortical amygdala. In this instance, you're looking at posterolateral cortical amygdala, as if I might know the difference. And what we observe, summarizing a great deal of anatomic analysis, is that in the cortical amygdala, the projections from individual glomeruli are stereotyped in nature. So some glomeruli will project more dorsally, others will project more ventrally, and this pattern of stereotyped anatomic projection appears to be maintained from individual to individual. We no longer see in the cortical amygdala this tight, insular, stereotyped segregation that demarcates individual glomeruli. Rather, individual glomeruli project to more broad and diffuse areas of cortical amygdala. Nonetheless, these projections remain stereotyped. On the other hand, in piriform cortex, we observe a highly dispersive projection from each glomeruli throughout the entire piriform cortex. If we examine the projections from 2 identical genetically marked glomeruli in different individuals, they are as similar to one another as the projections from different glomeruli. No amount of analysis, autocorrelation, cross-correlation, hierarchical cluster analysis discerns any spatial order in these glomeruli. In the piriform cortex, we observe this rich, dispersive distribution. And this has led to the suggestion by my fellows that perhaps the cortical amygdala is responsible for innate olfactory-driven behaviors, given the determined nature of these pathways, whereas this dispersive distribution, which has an apparent randomness, may be responsible for learned olfactory behaviors. And so Corey Root, in very recent experiments, has designed a paradigm to examine whether indeed the cortical amygdala can be implicated in innate behaviors. Now, remarkably, there are very few odors in the mouse, excepting putative pheromones, that elicit innate olfactory-driven behaviors. Very few, and even fewer, like none, in humans. But one odor, trimethylthiazine, the major component of fox urine, elicits robust aversive behavior. And Corey set up a 4-quadrant assay to demonstrate this. If you put a mouse in the middle of a very large 4-quadranted chamber, what you observe is that it spends 25% of its time in each of the quadrants. If, however, you put fox urine in one of the quadrants, then it avoids it. And you can see this sharp distinction between 25% of the time in the control case and about 3% of the time in the presence of TMT. And so he evolved this into a reasonably robust assay and, for simplicity, converted time spent into a aversive performance index. So we have a good assay, behavioral assay, for innate behavior. And then Corey simply designed a very simple experiment. He put Halorhodopsin, that was kindly provided by Carl, into the olfactory bulb such that it was robustly and diffusely expressed. And you can see that here. It's expressed in the mitral cells, the projections of the mitral cells, and the projections of the mitral cells to the cortical amygdala, to the piriform cortex. And he simply asked, he activated Halorhodopsin and therefore silenced not the bulb, but the individual projection loci from the bulb, and asked which of those regions, if silenced, could eliminate innate olfactory behavior. And it works. So if you put Halorhodopsin in the bulb, you see projections to cortical amygdala. If in this assay you now inactivate the axon terminals in the cortical amygdala, then what you observe quite consistently is you eliminate aversion to TMT. If you put the virus and Halorhodopsin directly into the cortical amygdala, as you might expect, you get the same result. And so, indeed, eliminating the cortical amygdala eliminates innate aversive behavior. This does not say the cortical amygdala is the only region necessary for innate behavior, but it, in fact, is necessary. Elimination of the cortical amygdala eliminates innate behavior. And we've done a number of controls, some of them actually in the last few weeks. If you perform the same experiment and ask whether inactivation of the cortical amygdala eliminates learned olfactory behavior, you do not see that. So moreover, in a series of controls, we can demonstrate that other projection sites, like the tubercle, do not seem to play a significant role in innate behavior. So it seems that at least innate aversive behavior is mediated by cortical amygdala. Now, let's turn to the more elusive problem of the piriform cortex. Recall this diffuse distributive representation, which led us to suggest that indeed the piriform cortex was required for learned olfactory behavior. The first question we needed to ask is, as we did in the bulb, what is the functional representation of this anatomic picture? And so very quickly, Dan Stettler developed imaging procedures. The piriform cortex is extremely difficult to access, but Dan was able to image piriform. And what he observed, recalling the distributive anatomic projections, what Dan observed were three important things for this discussion. First, if we look here at active neurons in response to three different odors, we observe that every odor activates between 5 and 15% of the neurons. Second, this immediately implies if every odor activates, say, 10% of the neurons, given the vast array of odors in the universe, that an individual neuron must respond to multiple odors. And this is indeed what we observe. In red, we observe the response to octanol, and in green, the response to 4 additional odors. And what you find is that indeed neurons respond to multiple odors. And the response of a given neuron to an array of odors seems to demonstrate that neurons themselves, responsive to, say, an aliphatic aldehyde, are as likely to be responsive to a different aliphatic aldehyde as they are to a benzene ring. And so there is what we call a lack of continuous tuning in an individual neuron. Moreover, the significant overlap that we observe implies that there is no cell in piriform cortex which in fact is highly specific. There is no grandmother cell in piriform cortex. Rather, the quality of an odor cannot be encoded by an individual neuron, but rather an ensemble of neurons. And finally, what we observe if we look over broad swaths is that, again, unlike other sensory cortices, neurons with similar receptive fields, with similar response properties, do not appear to be clustered. Rather, they are interspersed. So what I have told you or described to you is a piriform cortex which exhibits rather different character than the low visual cortex the low visual cortex or auditory cortex or somatosensory cortex. In these cortices, let's stay with the visual cortex, individual neurons are continuously tuned. A neuron tuned to an orientation of 90 degrees will fire less well at 80, 70 on out. And moreover, neurons with similar receptive fields are clustered. We do not observe this in piriform cortex. And in fact, the data that we observe is consistent with and can be readily modeled by arguing that an individual neuron in piriform cortex receives input from a random combination of glomeruli. In fact, recent data from Mike Ehler's lab seems to indeed demonstrate this, that a given piriform neuron appears to acquire about 1 to 200 random glomerular inputs. Now, the implication of random input to an individual piriform neuron is that within an individual, a given odor will activate a unique ensemble of neurons distinct from and discernible from the pattern observed with other odors. And moreover, that pattern will be persistent within that organism. But if indeed there is random input, this implies that the patterns will differ in every organism. So the same odor will give a random distribution of active neurons in different organisms. And the implication of that is that the patterns of neural activity can have no meaning. They can have no meaning, and meaning must be imposed by experience. And so another student in the laboratory, a postdoc in the laboratory, Gloria Choi, designed a series of experiments that I like to test this question as to whether an individual ensemble of neurons can elicit either aversive, appetitive, sexual, or social behavior, the same ensemble, depending upon learning. And so Gloria, in collaboration with Dan Stetler, I should point out, I don't do anything. Nothing. This is Gloria and Dan. In fact, many of you know from last night, I'm a terribly religious person. It's written in Leviticus that the work of the righteous is done by others. Susumu, you aware of that? So Gloria used virus encoding channel rhodopsin to create optically excitable ensembles in piriform cortex. And you will give me that infection with virus, in fact, will result in a random ensemble of neurons. And indeed, what she observes is that if you shine light on a naive ensemble of neurons in piriform cortex, you elicit no behavior. The animal exhibits no apparent recognition of this excitation. But if you pair light with shock, you generate robust aversive behavior. And we've done this in a number of different ways with a number of different controls. If you pair light with shock and then give light, the animal desperately attempts to escape. And this behavior is quite robust after a relatively small number of training sessions. We can do the same thing with appetitive behavior in this go, no-go licking assay, which I will not describe. What we observe is that if we pair light with a water reward, that indeed what we observe upon administration of light alone after a training paradigm is that a random ensemble can now elicit aversive, appetitive, and in a nice assay, social behavior. In this assay, what Gloria did was simply to take a male mouse, put him in a central compartment. On his right was a female mouse, and every time he went into that compartment, he was administered light, activating these neurons. And in fact, if you now take the female away and continue to shine light, activate these neurons in the compartment that once held a female, this male will essentially spend his life in there expecting the presence of a mate. And so indeed, random ensembles have no meaning when naively activated, but after learning, they can elicit aversive, appetitive, social, sexual behaviors, each dependent upon learning or experience. We can demonstrate, quite interestingly, that the very same ensemble of, say, 1,000 or 5,000 neurons in cortex can be entrained to elicit appetitive behavior, and then we can retrain the animal to make him think the activation of this ensemble will lead to shock, and the same ensemble will lead to aversive behavior. So the same random ensemble can be entrained to elicit different behaviors dependent upon learning. And finally, we can use chicanery and create two different ensembles with independent light sources, and we can entrain one ensemble such that the animal will run because it is shocked, and a second ensemble, the animal is shocked if it runs, so it will stay, and we can essentially make the animal dance, depending upon how we entrain each of multiple ensembles in piriform cortex. And we can do this across the piriform cortex. We've done it now at about 18 discrete loci running across the piriform cortex. So what this says is that indeed an apparently random ensemble of neurons in piriform cortex can be entrained to elicit behavior dependent upon experience. The position of the ensemble in piriform cortex is not restrictive. Wherever we introduce this ensemble, we can indeed elicit all behavioral types. And this implies that the piriform cortex, which can indeed elicit learned behavior, does not use space to map the nature of that behavior. This now poses two questions. First, if there is indeed reinforcement, learning imposed upon a random ensemble of piriform neurons, how is this learning, this reinforcement, physically manifested and imposed upon these neurons? And second, once an ensemble of neurons is reinforced, is strengthened, is afforded coherence, how is the behavioral specificity imparted? For time's sake, I will not discuss our thinking on how the learning is physically manifest or reinforced, because it's only a thought. But let me describe some very recent experiments that Gloria and Dan have done, which seem to indicate that behavioral specificity might be a function of where the piriform neurons indeed project to. The piriform neurons project to a number of regions. So if you put AAV encoding channel rhodopsin and some beautiful color, you can follow the projections from piriform, and they go to prefrontal cortex, tubercle, entorhinal, cortical, amygdala, among many other areas, hippocampus. Okay, so what Gloria did was a very simple experiment, and she simply created an ensemble and entrained with light and an appetitive stimulus in piriform, and then came back and tested by simply activating the axons of these piriform neurons in a variety of different regions. Here we'll restrict ourselves to cortical amygdala. And she can, in fact, recover all of the entrained appetitive response by activating the axons of piriform neurons in the amygdala. So the projections from piriform to cortical amygdala appear to carry information capable of generating appetitive behavior. We know this is not the consequence of antidromic activation because we simultaneously put in a virus encoding both halo and channel, and we use halo to activate the cortical amygdala, and channel to activate the cortical amygdala, halo inactivating piriform. Carl told me how to do this. Okay, so it seems then that a random ensemble of neurons artificially generated in piriform cortex is capable of eliciting any one of a number of different behaviors dependent upon experience, and we can begin to identify how behavioral specificity is imparted on the exact same ensemble of neurons. So what have I told you today? Just let me recapitulate. What I've told you essentially is that the brain is abstracting information in the sensory world. You knew this because in the sensory world, you're looking at wavelengths of light or chemicals, and you're transforming that information into spikes. That, by definition, is an abstraction. And so I would argue that what the brain is doing is not dissimilar to what the progression of art has done. So if we look at this now lost Michelangelo painting of Leda being seduced by Zeus in the form of a swan, we all recognize this, know what it is. But if you now look at this abstraction of Leda and the swan, what Cy Twombly has done was essentially distort line, form, color into a total abstraction. But I am as capable, having learned at MoMA that this is in fact Leda and the swan, I am as capable of recognizing this Twombly as Leda and the swan as I am the Michelangelo, despite its figurative and representational character. And what I've tried to argue is that the brain is doing precisely this. It is taking physical properties in the external world, wavelengths of light, frequencies of sound, chemical structures, and transforming them, abstracting them into an image represented by what appear to be a disordered array of active neurons whose meaning is only imposable by experience. And the olfactory system is an extreme example of this, but I think you might agree that hippocampal place cells, which bear no resemblance to the physical order of the external world, provide yet another example. And I would further argue that as one moves to higher visual cortices beyond V1, that one is going to see the same thing, that is a dispersal and randomization such that order is imposed by experience. Thank you.
Can we take questions? Questions? Barbara?
Richard, have you looked at the development of the piriform cortex? I would, synaptically, I would predict that some of the functions of plasticity change at a different time in that region than others.
Yeah, we have not. I would be very surprised if I saw order early and disorder late.
No, I'm not talking about that.
Yeah.
I'm talking about random selection of connectivity and then assigning it later to
Yeah.
tune it.
Yeah, right.
And that was
Yeah, I haven't done that.
Question over here.
Yes, that was really terrific.
Why do you think so?
Yeah, great question. Because you were able to show a step-by-step. My question is, could you extend this to other sensory organs of the body, say for hearing, for a visually impaired person? Have you thought about the possibility that it may be a similar mechanism, although maybe a different route?
Well, you know, my visual colleagues find it difficult to accept that. But if you look out in the room, you're looking at millions of images. It's inconceivable that you would have hardwired in your brain ensembles that in fact represent each of the million images we perceive upon looking out. And you know, virtually all visual information or visual knowledge is learned. And so I would argue, as you move to higher, perhaps infratemporal cortex, that you're going to see a picture more like what you see early on in olfactory cortex. The reason you see it, I believe, early on in olfactory cortex is that the early cortical stations for visual processing are extracting features out of a visual image. They're feature detectors. They are not truly encoding the image. And these features then have to be reconstructed in higher visual centers. And it's this reconstruction that I think is going to involve randomness.
Question? Marion?
How stable is the order imposed by experience in the experiments that Gloria performed? Did she ever try and reverse the experience paired with the stimulation?
Yeah. So I showed one experiment where she can reverse it. I mean, firstly, the learning lasts for quite a long time, months. But you can reverse it by changing the paradigm.
So I have two quick questions. One is the number of neurons that you induce for your random ensemble. Is there a discrete number that you need? Is there a minimum and a maximum, or is it continuous?
So we've studied that, And you begin to, a given odor activates 100,000 neurons in piriform cortex. We begin to be able to entrain and elicit behavior with infection of 200 to 300. And by a few thousand, where we've maxed out.
Smells that generate innate behavior, but we all know that there are smells that inherently we don't like or we do like. And so how do you explain that in terms of randomness?
Yeah, there are two ways to explain that, and we could probably talk about it later. The first way is a cop-out, and I like those. You argue that these perceived odor qualities, these categories, are encoded in the cortical amygdala. Okay, that's a cop-out, I think. But we can talk later. We have with Larry Abbott modeled categorization from randomness. And it works because if the output of the piriform cortex is also random, then some of the output neurons, the neurons that receive input from the piriform, will get more inputs from different categories. And you can use those for categorization. I would argue that you can categorize whether you, that may be imposed by experience.
So are your glomeruli the equivalent to the color receptors in the eye?
They are feature detectors, and if you want to restrict vision, if you want to look only at color vision, then the individual odor receptors will be equivalent to the color receptors.
So the number of glomeruli is sort of the space in which this thing is taking place.
Yes, but you use them in different ways.
No, I understand
The vision you comparate, here you don't.
Okay.
Okay, I think I'd like to ask the last question. And as you're thinking about more complex odors, so you think about mixtures of odors, and if you were to look along the sensory representational pathway, different parts of piriform cortex, how do you think the representation would reflect changes, perturbations, rather than recognition of a particular odor, detecting changes or variants?
You mean like mixtures? Or?
Mixtures or sort of subtle variants, individuals that have, let's say, an enhanced capacity for olfactory discrimination, not just detection.
Yeah, the latter I can't comment on. People have looked in strains of dogs to ask, how do we explain the enormous sensitivity of a bloodhound's nose? There's no apparent anatomic difference. The first question is a more subtle question, and that is, what happens when you have mixtures or change concentration or entrain to a mixture and then give only a component of the mixture? We know a few things. We know that if you give odor A, you activate 10% of the neurons. If you give B, you activate 10% of the neurons. C, 10%. If you give A plus B plus C, you activate 10% of the neurons. So there is strong suppression and the introduction of new cells that are A plus B responsive. And I didn't have time to talk about the mechanisms of suppression. There's a huge recurrent network with global excitation and scaled inhibition, local inhibition.
Great. Well, let's thank Richard again.